Symposium Organizing Committee member Steve Anderson.
Hello, everyone. We hope that you had Is this the one? Okay. So we hope you guys had a good lunch and you enjoyed it. First, we would like to remind you that tonight, following the symposium, we'll be having a social event at 6 p.m. at Cafe Excel. And that's in Bryan, Texas on Texas and University. So there will be pizza and appetizers provided free of charge, and there will also be a cash bar available for you guys to come socialize and get to interact a little bit more. So we will be at this event. We will be having the presentation of the winners of the poster session, as well as acknowledging several of the traveling oral speakers, as well as the Texas A&M speakers. So it's a great opportunity for everyone to come during the evening and meet and socialize with keynote speakers and the rest of the general audience in the crowd. So we hope that you come. We invite you to bring your significant others along with as well and come have a great night with us. Okay, so now we will hear from our third keynote speaker of the day, Dr. Kirti Rathore. Dr. Rathore is professor in the Department of Soil and Crop Sciences at the Texas A&M University. I don't have it. Sorry. So he is a professor in the Department of Soil and Crop Sciences at Texas A&M University. His research focuses on genetic improvements of crops through the development of protocols for efficient delivery of genes, optimal expression of transgenes, rapid recovery of transgenic cotton, rice, and sorghum. His projects include regeneration from cell and tissue cultures, use of new reporters and selectable marker genes to understand and improve transformation processes, promoter analysis, enhancement of disease resistance in plants, conferring drought tolerance in crop plants, conferring insect resistance in crop plants, improving nutritional quality of seeds, and production of recombinant antibodies and vaccines in plants. His lab has recently applied for deregulation of a transgenic cottonseed with localized suppression of gossypol, making his lab one of the handful of public programs that has filed for release of a deregulated transgenic crop in the public sector. Additionally, his group is working on root rot nematode resistance and development of CRISPR-Cas protocols for cotton. Dr. Rathore holds a BS in zoology, botany, chemistry, and a master's in plant science and a PhD in plant physiology. The title of his talk is From Conception to Towards Deregulation of Ultra-Low Gossypol Cottonseed, a 20-year odyssey. Please help in joining Dr. Rathore.
Thank you for the introduction, Steve, and thank you, the organizing committee, for selecting this particular talk, which is my favorite talk for the last 20 years or so, and it's been a long journey, but at least as far as taking an idea to the product stage, I think we're pretty close, hopefully. And also, thank you for sticking around after lunch. Usually, once people have had their lunch, they leave. So thank you for sticking around. Now, this is a subject that many people who are not in the cotton area would know little about. So I'll have quite a few introductory slides, so to give you some background as to what the problem is and why I got involved in this and why we spend so much time on this thing. So if you're like me 20 years ago or 22 years ago, you wouldn't think that cotton plant produces almost 1.6 times more seed than it produces fiber in terms of weight. So overall, worldwide, we are producing almost 24 million metric tons of fiber. Along with that fiber, we produce about 47 million metric tons of seed. Now, cotton seed has about 22-23% protein. So that means that in terms of cottonseed production, we're producing about 10 to 11 million metric tons of protein every year. And it's a good quality protein. Now, a human being requires about 50 grams of protein per day to survive. Now, if you could utilize that protein directly for human nutrition, you could meet the basic protein requirements of anywhere between 550 to 600 million people. And that's a lot of protein that is right now not being utilized in a very efficient way. And the main reason for that is that cottonseed contains this compound, terpenoid gossypol, and gossypol is toxic to monogastric animals, including human beings. And so this is this was one of the impetus for us to get started on this project. You may be looking at this little mountain. It's cottonseed just piled up right outside Lubbock. And just to get some perspective how big that is, here is a pickup truck. You can see that. So there's a lot of cottonseed produced all around the world, and it'll be nice if we could utilize this cottonseed as a source of protein. So as I mentioned, the protein content of cottonseed is about 22-23%, and it's relatively good quality protein. And as I said, monogastric animals or human beings cannot use it because of the presence of gossypol. Now, the gossypol is produced and stored in these dark colored glands that you can see in the seed. This is a slice of a seed showing these glands. That's the structure of gossypol here. Now, as I said, gossypol is toxic to monogastric animals such as pigs, chicken, fish, and so on. The only animals that can handle gossypol, and that to a certain level, are the cows, cattle. And we do utilize cottonseed oil. So cottonseed is crushed to get oil. Oil is refined, and through some chemical and physical processing, gossypol is removed. And after that, it's pretty healthy, and we all eat cottonseed oil. This slide shows the countries, some of the countries that produce cotton. So here are the 4 major producers and their production levels as well as the export levels. Here are some of the African countries, West African countries that also produce cotton. And cotton is a big crop there. These numbers in the red really show the export income, that the fact that cotton export is number 2 earner of the export income in this country, Burkina Faso, number 1 in Mali, and so on. And some of these other countries in Africa that also produce cotton. But look at the fertility rate. These are the countries where the population is rising very fast. There is already protein malnutrition, and they're going to face much more severe problem as time goes. Now, a lot of these countries that produce cotton basically survive on some of these cereals. That's the staple food for them. And the level of protein that these cereals have is much lower than what you normally want in a diet. A lot of countries, people just survive on cassava or yam that have extremely low levels of protein. By the way, the project that I'm going to talk to you about today, Dr. Borlaug, he was in our department. He was one of the biggest supporter of my project. He knew the value of this project. U.S. farmers are also very interested in this particular product, or gossypol-free cottonseed. And in fact, a survey was done in 2015, and they put improved cottonseed value as number 4 item on their list of what they wanted. And obviously, if the cottonseed becomes a source of food or even as a source of feed for some of the monogastric animals, its value will go up, and cotton farmers will get more value for their money without doing anything additional that they do now. So as I said, we do utilize cottonseed oil in our diet directly, human nutrition. Also, so we're not throwing away the cottonseed. We're still using it before or after oil extraction for as a feed for cattle now, and we obviously cannot use it for some of these monogastric animals. So what's wrong with that? Somebody will say, okay, we're still using it indirectly for human nutrition. The fact is that cows are not the most efficient animals. So a milk cow produce, I mean, it has to, you have to give it 3.66 kilograms of crude protein to get 0.83 kilograms of milk protein. And the protein conversion ratio for beef cattle is 20, meaning that 20 pounds of crude protein to get 1 pound of beef protein. So very inefficient animals. Compare that to some of these other animals. So here is pig, chicken, 5 times more efficient. Eggs are the most efficient way to convert crude protein into animal protein. And some of the fish that we eat more and more, the aquaculture grown fish like salmon, tilapia, are also much more efficient than the cows. So if you had a limited amount of protein, it's better to give it to some of these animals than to just simply feed it to the cows. So the problem has been known for a long time, and people have been trying to address it for a long time. And in 1954, McMichael discovered a glandless mutant of cotton. Now, as I said, gossypol is present within these dark colored glands that you see all over the cotton plant. And since the plant didn't have any glands, it did not have any gossypol. And there was a great deal of excitement during that time, and a lot of national and international projects were launched to incorporate this trait into commercial varieties of cotton. And at the same time, there were also animal nutrition studies were done, and they found that the cottonseed, which was gossypol free, was good enough to feed to the chicken, pigs, fish, and so on. In fact, it was considered good enough for human consumption. In fact, Texas A&M tried to to introduce these roasted and salted cottonseed kernels. They called it tamu nuts, and people actually like the taste of those tamu nuts. Now, so this is the situation. Is so most of the cotton we grow today, wild type cotton, is glanded. There are glands present in the leaf and other parts of the plant, and here is the seed. The seed gossypol level is about 10,000 parts per million. Here is a mutant glandless cotton, so no glands in the leaf or any other part, certainly no glands in the seed, and very low or negligible levels of gossypol. Now, gossypol that is present, and this is a picture showing the presence of glands in the leaf, in the sepals. It's also present in the petals, flower petals, ball, here, bracts tissue, stem. Gossypol present within these glands protects the plant from insects, and there is some reason to believe that it also plays a role in defending the plant against certain fungal diseases. Okay, so it's necessary for the whole plant. So once the farmers try to grow this glandless cotton in their field, their glandless cotton was attacked much more heavily by these traditional insect pests of cotton. There are insect pests that normally do not go anywhere near cotton. They were attacking the glandless cotton. So for that reason, there is no large-scale production of glandless cotton anywhere in the world right now, despite the fact that it proved highly effective in terms of as a feed for monogastric animals, as well as a source of nutrition for human beings. So what is the desired trait in cotton? So you want cottonseed to be low in gossypol. Now FDA considers cottonseed products safe enough even for human consumption if the levels of gossypol are below 450 parts per million. And but you want obviously want levels to be low enough or lower than what FDA considers safe. At the same time, you want the normal levels and complement of these gossypol and other terpenoids in rest of the plant. So that was our goal when we started, and I started this project in '96, and we had to develop all the tools and technologies to conduct this project. So first, we obviously need an efficient transformation method. We need a target gene that we can silence so we can suppress the gossypol. We obviously need to do it in a seed-specific manner in order to make sure that the rest of the plant doesn't suffer from insect attack. And we needed a gene silencing mechanism. So we started this in '96, about '97, '98, we established this transformation protocol for cotton. And this was agrobacterium-based method. Just to give you some idea how difficult that is, starting with the transformation of the cotton discs or stem or hypocotyl tissue to get to this stage when you get a transgenic plant in the soil takes about 10 to 12 months. So that's the time frame. But we had a pretty good system working in the lab. We needed to have a good seed-specific promoter, so we went after the seed storage proteins. And globulins are major seed storage proteins in cottonseed, and we figured that the promoter that drives the gene for this particular seed storage protein should be seed-specific, but we had to make sure that that is the case. So we isolated the sequence of the promoter and hooked up with the reporter gene GUS-A and transformed those constructs into tobacco, Arabidopsis, and cotton, and examine the activity of the promoter. So we find that in the developing cotton embryos, the promoter becomes active at around day 16 post-anthesis. And this is the higher magnification image of the same embryo. As you can see, this blue coloration here, that means that the promoter is just becoming active. And as the embryo grows, the promoter activity becomes stronger and spreads throughout the embryo. By day 25, the promoter is active throughout the embryo. And this is just about the day when the gossypol production begins in the embryo. Okay. We also made sure that the promoter is tissue specific. It's not active in any of these other tissues. The only place it's active is in the cotton seed. So, and this is the gene we are targeting. I'll tell you a little more about that in a second. But there is a good overlap between the promoter activity and the activity of this particular target gene. So when I started this work, the only gene silencing technology that was available or was known was the antisense technology. So we used this technology and made several constructs. Okay, this slide is an important slide. So what I want to say here is that this is the gene we targeted, delta-cadenine synthase gene. Delta-cadenine synthase is an important enzyme that's involved in the conversion of farnesyl diphosphate to delta-cadenine. And some of these biochemical steps were known at the time, and this gene was the first one that was isolated. So we isolated that gene from Gossypium hirsutum. The other thing I want to point out here is that these two parts of the plant, cotton plant, like the flower petals and the seed, the predominant terpenoid in the glands of these two parts is gossypol. The green parts of the cotton plant, like the leaf, stem, ball, rind, and sepals, and so on, the glands in those parts contain not just gossypol, but also hemigossypolone and heliocytes, and they're all derived from the same basic biosynthetic pathway. Some of these compounds have much higher insecticidal activity than gossypol. So we made some antisense constructs, and this was our several lines we derived. And these are the Gaussy-Paul values in all these different lines. So these are T1 pooled T1 seeds. And we do find that some of the lines towards your right side, they are low in Gaussy-Paul. So this is in T1 seeds. And we thought, okay, that's great. Let's look at what happens to the T2 seeds. And when we checked the T2 seeds, we got a big shock here. So the bars here in the red color, they are the values for the T1 seed. The bars in yellow or orange color here are T2 seeds. So what that told us is that the trait is not stable. We got some reduction in Gaussy-Paul levels in T1 seeds, but not in the T2 seeds, except for one particular line here that showed us low levels and looked pretty hopeful. So we carried on up to about T4 generation with that particular line, and we did see some reduction in the level of Gaussy-Paul in the seed. The levels in the leaf were similar, whether it's wild type or the transgenic cotton plant. So with the antisense technology, we were able to reduce Gaussy-Paul in the seed to about 1700 ppm. So we came down from about 10,000 parts per million to about 1700 ppm. So we're nowhere close to what FDA considers safe, which is 450 ppm. So that was a big setback and almost 4 years of work. It couldn't even publish something like that. So we thought, okay, we will try. By that time, there were a few years after that, RNAi technology came along, and we thought, okay, can we utilize RNAi technology to do the same thing, which is supposed to be more powerful silencing technology. Now, before we even started our work on RNAi, there were several papers out there that showed that RNAi can spread from its point of origin to other parts of the plant. And they showed that in different ways, and probably no need to go into detail, but there were several papers that showed that if an RNAi occurs at in certain part of the plant, it can spread from that part to other parts of the plant. Now, if that was to happen in our case, where we are trying to suppress Gaussy-Paul in the seed, the RNAi would have spread to other parts of the plant, in the leaf and other parts, and it would have silenced the gene and no Gaussy-Paul. So we would be in the same situation as the glanless cotton. And in fact, some of the pioneers of this RNAi work, for example, Waterhouse. I think I lost the battery here. Okay. They wrote some reviews and talked about the fact that tissue and cell-specific RNAi may not be possible because of the spreading of silencing. Now, if we believed these papers, I probably wouldn't be standing here talking to you today. We went ahead and did the experiments anyway. And so again, the slide thing works, but this pointer thing is not working. Anyway, so basically we used the same promoter and we targeted the same gene, delta-cadmium synthase gene, and the only thing we did different was to use RNAi instead of antisense. Okay, next slide. Someone can. Okay, there you go. So we made we used about 600 base pairs of the trigger of the sequence as the trigger sequence. We made some hairpin RNAi construct. Next slide. Someone can. Yep. So we made the hairpin RNAi construct, and again used agrobacterium. Okay, thanks. And used the agrobacterium to transform cotton. these are some of our, this is the result from our T1 seeds. So there are several lines here. Okay, the pointer thing still doesn't work. Okay, just the slides. Okay, all right. Uh, yeah, so line number 232 and line number 81, these lines look pretty promising, and the levels of the seed gossipol in the pooled seed gossipol is shown. Yeah, sure. Okay, so we thought we'll go ahead and test these. So these are, remember, these are pooled T1 seeds. They would be segregating for the transgene. Okay. Oops, that'll do it. So we carried out southern blot analysis on these three lines, and we see that the transgene is certainly integrated into the genome. And this was our first result, which looked very promising. So here are individual 10 seeds from a wild type or non-transgenic plants, and these are the gossipol values in individual seeds. These are the two lines, line number 2 and 32. The seeds that had the gene, which is shown by this PCR result, had very low levels of gossipol. The seeds that did not have the transgene have normal levels of gossipol. So remember, these are T1 seeds that are going to be segregating for the transgene. So that was very promising result. And if you look at the HPLC chromatogram, normally the cotton seed that will show a big gossipol peak like that. And here are some of the seeds that are transgenic or expressing the RNAi construct. Can barely see the gossipol peak. Okay. And we obviously checked the levels of transcripts, and we showed that the gene that we were targeting was indeed silenced in these two transgenic RNAi lines. We confirmed that again with northern blot analysis, and they showed the same result. We've done the enzyme analysis for this delta cadmium synthase enzyme, and the enzyme levels are extremely low in the RNAi lines. So these are all, these results are from the embryos, developing embryos. So far so good. What happens to the leaf and other parts of the plant? And so we chose the same two lines, line number 2 and 32. Again, we chose 10 plants from, these are again T1 plants, so 10 plants from each, in each case. And these are wild type plants. Now remember the slide that I showed you earlier, that the green parts of the plant contain not just gossipol, but also hemigossipolone and heliocytes. So these are the values for gossipol, hemigossipol, and heliocytes. And as you can see that we don't see any reduction in the level of these terpenoids in the leaf. Here are some other parts that insects normally like to chew on, and the terminal bud, the floral bud, bracts tissue, petals, ball, and the root. Again, the red bar is wild type or non-transgenic cotton, and the two green bars are the two RNAi lines. And we see that the levels of these terpenoids are very similar, whether it's transgenic plants or the RNAi plant. So this these results show that the trait is totally tissue specific, that the reduction of gossipol we are finding is only in the seed and no other part of the plant. So we went to T2 generation to make sure that the trait was stable. And so this again, line number 2 and 32, these are T2 seeds from T1 homozygous plant. And all the 50 seeds that we tested have very low levels of gossipol. So this result showed us that the trait is stable and is transmitted to the progeny. So unlike the antisense work that I showed you earlier, which was not stable. Most of the lines reverted back to normal levels of gossipol. This looked like it is promising. This was another third line that we had tested. Again, these are individual values of gossipol in these seeds. And if you average those out, it's about 190 parts per million gossipol in those seeds. And the wild type seeds have close to about 10,000 parts per million gossipol. So what happens if you take these lines to the field? So all this work that I had shown you earlier was done in the greenhouse stage. Things are different once you go to the field. We wanted to make sure that the trait was stable under field conditions. And so we have three different lines that that we had selected and tested those in field conditions in 2009, 2010, and 2011. And again, as you can see, compared to the non-transgenic, the levels are low. Seed gossipol levels are low. So the trait is stable under field conditions. So that's where we are. So we are now calling this trait ultra-low gossipol cottonseed. We were able to reduce the levels from somewhere around 10,000 parts per million to about 250 parts per million. So if you go from generation to generation, it's not going to be exactly the same number. It may be 200 sometimes 275, but overall we have about levels about 250 parts per million, which is way below what FDA considers safe, and also WHO, FAO. Their numbers are much higher. They're about 600 parts per million. Being safe for human consumption. But the main important thing is that the foliage and floral parts of the plant contain normal levels of gossipol and related terpenoids. So their defensive functions should not be compromised. So starting in 96 to about 2006, when we had our first publication on that, this was the situation. So this is again just to kind of reiterate what I said. This is commercial cotton, 10,000 parts per million of gossipol in the seed. Here is the leaf, and you can see the glands clearly. This is the glandless cotton, very little or no gossipol in the seed or any other part of the plant, and no visible glands. And this is our engineered cotton, ultra-low gossipol cotton seed. Values in the seed are about 250 parts per million, but the leaf and other parts of the plant have normal glands and normal levels of gossipol and terpenoids. So it looked very promising and looked like the trait is stable and it is inherited generation after generation. So in 2006, we started taking some steps towards deregulation of this trait. And we generated many more lines. We certainly cannot match what all you guys in DuPont Dow do, who create something like 1,000 lines or 2,000 lines for any given trait. But we were able to generate at least 300 or so lines. And from those, we checked them for number of copies. We did a lot of southerns and insert localization, silencing, made sure that the gene was silenced and the levels of gossipol were low and the trait was stable in field conditions and so on. Carried out a lot of biochemical analysis. We did inheritance analysis. Over 2014, 2013, 14, and 15, we carried out multi-location field trials in 3 different states. There were 8 different trials, and we looked at the agronomic performance of a couple of lines, and also we did the seed composition analysis on those. Obviously did the fiber quality analysis, and all these analyses showed that apart from the low level of gossipol, everything is very similar. And in September, October of 2017, we filed a petition to USDA APHIS and also submitted a dossier to FDA. Okay, so that's where we are, and maybe I shouldn't, you can all check the timeline, already gone through that. Okay, so here is where you can use this cottonseed. You can use it to feed these animals, which are normally very susceptible to toxin, or you could even use it directly for human nutrition. And aquaculture, which where I think that it could really meet the demand for aquaculture feed, because it's one of the fastest growing industries. It's growing at a rate of 8% per year. And there is already shortage of feed for the aquaculture species like salmon and tilapia and shrimp and so on. Skip that slide. Probably, yeah, this is just a very quick slide to show chicken meat production in India. India used to be a largely vegetarian country, and look at the level of the chicken production, almost nothing until about 81, and now it's skyrocketing. And the same sort of thing is happening in a lot of other poor countries where the middle class is rising and there is much more demand for meat and so on. Okay, yeah, just the last or second last slide here. So now every time you do something, the question comes up, can you use CRISPR to do the same thing? Well, CRISPR, okay, if you were to target delta-cadmium synthase gene with CRISPR, there is no way to do that. We find that in the embryo there are at least 10 genes that are expressing delta-cadmium synthase gene. It's a member of a large gene family. Okay, so that part won't happen. Even if you can find a single copy gene in cotton that you can target to do the same job, which by the way we do have a gene that we think will be very good for doing the same thing. But you can target it, but you cannot make it seed specific. If you knock it out once, it's gone forever. Okay. Now, there is a possibility that one can use CRISPR interference to do this work, but again, your plant is still going to be considered transgenic, so you really don't gain anything by using CRISPR interference. So here are the countries where there is rampant malnutrition, all the countries that are shown in color here. Here are the cotton producing countries that suffer from malnutrition. So these countries can benefit a great deal from this particular product if they adopt it. And here are the people who actually did the work. So there are people in my lab, most of them have left, but the ones in yellow that are shown here, they're working on this project. We had a good collaboration with Dr. Wayne Smith and Steve Haig that are in our department. And one of the big thanks is to USDA ARS people, Bob Stepanovic and Lorraine and Al Bell. They were extremely helpful to us in terms of analyzing gossypol in the seed and other parts of the plant. Couldn't have done this work without them. And since 2006, we had a big support from Cotton Inc. to carry out this project. And there are some of the other collaborators. In fact, Sam Reddy is in Dow right now. And he was involved in the early part of the promoter isolation and so on. So, and these are people who are contributed funding. And with that, I will stop and be happy to answer any questions.
Okay, thank you, Dr. Thor, for sharing your story with us. So we have a few minutes for questions. Remember that if you have a question, please raise your hand and we'll bring you a microphone. So I guess we can start with Julian.
Do you know if this gospel is only de novo synthesized in seeds, or can it get transported?
the seeds are, the gossypol that's present in the seed is produced in the seed. But it does other parts of the plant. Each and every cell is capable of producing gossypol. A lot of times, gossypol and these other terpenoids, it comes in response to attack by insects and so on. Insects or some disease, fungus or some other disease. But the gossypol that's produced in the seed is coming from the seed, is produced in the seed itself.
Flora and topology path. Right?
No, we are not lowering the levels of gossypol in foliar tissue and the leaf or anywhere. That was the slide that I did show.
Okay, so my understanding, you can reduce the gossypol level in the condensate, but the leaf still has some high level, lower level.
It's the same level as the parental plant in the leaf.
So I thought you can keep the foliage or the floral parts at a normal level to protect the plants against the insects. So if you can separate them, so my question is they controlled by the different genes or the same gene control?
Sorry, I don't fully understand your question. So the seed gossypol levels are the only ones that we have been able to reduce. All the other parts of the plant have the same levels of gossypol as the parental plant. So we have not either lowered or raised the levels of gossypol and terpenoids in other parts of the plant. So it's highly tissue-specific trait.
Excellent presentation. The question I have is if the human being consumes on a constant basis this source of protein, what are the chances of getting toxic if they consume and if they exceed the limit of the FDA regulation?
Yeah. Again, I did point out that you can use this for human nutrition. Personally, I think that is a much better source of. It's better to use this as a feed for animals, aquaculture species, and poultry, for example. As I said, there were a lot of studies were done in 60s, 70s, and that's where FDA determined this level of 450 parts per million to be safe even for human consumption. So nobody is going to eat this day in, day out, right? Or whenever people use it, it'll be used as a supplement to whatever else they're eating.
Right. Dr. Rathore, we have a question from the webinar from Colbin Joy. Right. What do you believe is the biggest barrier to commercialization? Why has it not been picked up by the industry?
Maybe these guys can answer that. No, I'm joking. Well, I would be the happiest person. Or 10 years ago, we knocked at the doors of Dow, Bayer Crop Science, Monsanto, and well, obviously we were not successful in persuading them that this is a worthwhile technology. I guess this is my, and maybe one of you guys can answer later on. I guess they did not see how they can recover the investment by investing in this technology, and it is a big investment.
So I guess this is a follow-up to what Dr. Patel was asking. So how is this gossypol metabolized in animals or humans? Like, is it broken down, or if you keep consuming it, does it accumulate like on the skin or any other body parts?
No, it'll get metabolized. It'll get broken down. I mean, again, there is a limit. Everything is there are a lot of toxins we consume, basically, but I mean, your body does break it down, and I think all these things have probably been looked at by FDA earlier on, many decades ago. By the way, I was talking about aquaculture. We have done 2 aquaculture feeding studies. We have fed this cottonseed to shrimp, and they grew quite happily and no problem. In fact, and this was done in Corpus Christi with some of our collaborators. We brought some shrimp back here. We analyzed the shrimp again with the help of our collaborators, Bob Stepanovic in his lab. And that shrimp that was fed on the ultra-low gossypol cottonseed, we saw no traces of gossypol in that shrimp. So it's safe enough for people to eat, even without removing the vein, which normally people don't remove that. And so even then, we could not detect any gossypol. So what I'm trying to say is that people do not have to eat cottonseed. The other study that we've done is on southern flounder. So that study also found that it's a very good source of protein for southern flounder.
I have another question from the webinar from Benjamin Merritt. Have you looked at the mechanism that is possibly causing the RNAi from translocating to other parts in the plant? In your case, as opposed to other cases where it did.
Yes, yeah, no, we have spent a great deal of effort trying to figure out, well, whether that was the case or not. And some of the studies that I did not show here, they were done using this grafting technique. So they had a silence plant, and they took a stalk of that, an unsilenced scion or something. They did a reciprocal grafting and showed that the silencing moves up or down. We've done exactly the same thing in cotton. We do not see any sign of spreading of silencing. The other way to look at this, or indirect way of making certain that that does not happen, is that, and I showed you one of the slides where we were looking at the segregating generation T1 seeds, and the seeds that did not have the gene were showing normal levels of gossypol. So if the RNAi was to spread, it would have spread from those silent seeds to these null segregants. And it did not. And we've done that in many different ways. So we don't know why cotton is different, but the early studies that showed this kind of spreading were done in tobacco and in Arabidopsis. So we're lucky, I guess. But having said that, we also try to repeat the tobacco studies or grafting. We did not see spread of silencing in tobacco.
I personally have a question,
Sure.
just to tie this together to the theme. If a student came to you with a similar, and I know you face some unique challenges with this project, but if someone came to you with a similar idea in this day and age, what would be kind of your advice at this point? Your 1 minute answer to that.
Advice in terms of what? Whether to do something like this, or
What kind of impetus do you need to, from the industry level, to motivate a project that's going to end up being some like very large and possibly risky project?
well, okay. If I did not see the implications, and it's huge implications, 500 million people, so protein source for 500 million people. If it didn't have that sort of impact or potential impact, we haven't impacted anything yet. If it didn't have that sort of potential impact, I probably would have, once we published the paper, we've moved on to something else. Having, as you probably can tell, I come from India, spent about 21 years in India, and I've seen poverty firsthand, and I've seen hungry people. I've seen people, who live on just bread alone, and so I've seen how people suffer from this protein malnutrition. Also, my father was a doctor, so he was seeing all these patients. A lot of the sickness comes from being hungry and not having enough nutrition. So maybe I'm a unique case in that respect, that I didn't give up. Many times I wanted to give up, but and uh, it's again, this is science, and you have to be a little bit of stubborn to be able to accomplish something. There is a very fine line between being totally foolish and being stubborn and all that. So uh, we I was pretty hopeful that we'll be able to do it, and we could have done it sooner if we had funding right from the beginning. We just didn't have funding. In fact, there was about a period of 4 or 5 years we had no funding to do this work, and so during that time, that Dr. Borlaug also tried to help us, called some Rockefeller Foundation and so on, to say, okay, can we get funding for this? And obviously we didn't get any. So that's why it took a little longer than it should have done.
Okay.
Thank you.
This one might not be working. This one's not. Is this one on?
That one's on.
It's not on, is it? Okay, can you guys hear me? Okay, perfect. So next we'll be hearing from our fourth. Excuse me. Into the mic. Okay, here we go. So next we'll be hearing from our fourth speaker, Dr. John Van Hermit. Dr. Van Hermit held
testing all the way from prelim testing to advanced testing across the entire northern region of the U.S. And I'm very proud of the team that we have up in Minnesota running this program, and we have a lot of great funding from commodity boards and government and state sources. But prior to all this, I was at the University of Nebraska, and I worked on maize at the University of Nebraska. And I worked specifically on maize quantitative genetics and breeding technology at the University of Nebraska. And these are some of the people that worked with me at the University of Nebraska. Several of my graduate students at the University of Nebraska just recently finished. And so what I chose to do today was to discuss some of the recent results obtained by some of my recent graduate students who graduated from Nebraska, rather than talk about a bunch of soybean projects that are currently in progress. So I thought this was a good point of things that are complete, but not too old. So that's what I'll be talking about today. So even though I currently work on soybean, I'm going to be talking about corn. So there I am, again, to recap, maize first, born and bred academically in maize, and now I work in soybean. And I'm probably the worst crosser of soybeans out there for a professor of soybean breeding. I'm terrible at crossing. You don't need to do your crossing for me in soybean, but I'm trying to get better at it. All right, so this is where we're going here. We're going to talk about a little bit genomic selection in general. I'm going to give an introduction, a little background, and some uses generally in plant breeding programs. And then I'm going to talk specifically about the genomic prediction breeding program that we looked at for single cross prediction in maize, okay, and several specific aspects related to that. Okay, so this is a breeding pipeline. This is the soybean breeding pipeline that I put up, okay? And the goal of this breeding pipeline, of course, is to develop superior varieties and superior parents. And if we're effective at executing this pipeline, like we hope to be, then we're going to make gradual genetic gain, okay? The populations, the parents, the new cultivars that we develop from that pipeline are going to be gradually a little bit better than the ones we had before for highly quantitative complex traits. And although it can be quite slow over long periods of time, you'll see that, for example, in soybean, we produce soybeans now that have twice the yield potential than they started with back in the 1930s and 1940s. So it's slow, but steady and effective. So again, how can we make that breeding pipeline better? All right. So when I'm in the field doing my crossing, thinking about how can I make this cheaper? How can I make this faster? How can I make this more accurate? How can I find more useful genetic variation? These are basically the breeder's equation or the genetic gain equation boiled down into words. Okay? I'm also thinking about how tedious this is and how I wish I was pollinating corn plants at that point in time, but that's another story. All right, so two current breeding methodology research areas that are very popular these days include genomic selection, okay, of course, and what we've heard a lot about today is high throughput phenotyping. And really, if these things are employed and applied directly, usefully to a breeding program, what they should allow us to do is to broaden that funnel of a breeding program, allow us to cram more germplasm through that breeding pipeline for the same cost, or hopefully maybe less cost. And if we can do it faster, of course, we're going to shorten the length of that funnel, okay? So there's all kinds of fantastic things going on in high throughput phenotyping. We've heard a lot about those today. We have a few things going on in our lab. I'm going to be talking about genomic prediction and selection today. All right, so why do genotyping? We want to do genotyping to more effectively and efficiently, hopefully, identify or measure genetic value for individuals, okay? So if you think about a single marker here, okay, of course, we have a causal polymorphism here. We want a marker here that's closely linked to that causal polymorphism, so we can use that causal polymorphism, or the marker, basically, to differentiate our individuals just at that one marker data point. All right. So if we can do this effectively, of course, we can perform selection during the off-season. We can increase our selection accuracy, possibly, and we can increase the selection intensity if our genotyping is cheaper than our phenotyping. All right, and this works really well for really simple traits. All right, so I know now that I've run the soybean breeding program in Minnesota for the last few years that we have really good markers for SCN resistance. There's nothing I love more than anything is taking 10,000 plant rows and say, genotype all these things for RHD1, and I can remove a whole bunch of them just based on that marker. That really makes my job a lot easier. We also have good markers for aphid resistance, good markers for fatty acids. They really make my job easier. Yeah. So for simple, relatively simple or very simple traits, this works really well. But the problem is that a lot of traits that we're interested in are highly complex. All right, so this is an example out of Holstein where we have a relatively simple trait here in terms of fat percentage in the milk. Most of the variation for this trait is controlled by a very large effect QTL here, and then a couple of more minor effect QTLs. But oftentimes we're interested in highly complex traits like overall type, where there isn't a single gene that controls most of the variation. All the genetic variation for this trait is coming from sort of a uniform signal across the entire genome. So, example here published several years back now in flowering time by the Buckler lab showed that flowering time, even though it's highly heritable, a variation amounting to a month or more in flowering time is controlled by the accumulation of many small effect alleles, not even the largest of which I think was about a day in flowering time, all right? So I think we knew this for a long time, and then we sort of rediscovered this recently with a lot of genomics and GWAS applications. But we're kind of back to that paradigm. So if this is really the case, then you look at classical studies, well, I guess not really classical, but almost classical studies, back comparing phenotypic selection to marker selection. You can see that this is the case when you just use regular traditional marker-assisted selection, that they're not going to perform very well when you compare it to phenotypic selection, if that is the case. So there's this new paradigm, well, in plant breeding, about 10 years old now, I guess, genomic prediction and selection. And the idea is rather than go and find individual markers to make selections upon, okay, we actually skip that part. We do no QTL mapping. We do no significant testing. And rather what we do is we take DNA marker data across the entire genome. We combine it with phenotypic data, and we call that the calibration set in order to calibrate or train a model. Sometimes it's called training population. Sometimes it's called calibration set. All right, so we train this model using all the genome-wide markers, and then we simply use those predictions, okay? Apply those to, we apply this model to a set of selection candidates that have been genotyped but not phenotyped, and then we use those predictions to make selections. And again, there's no testing of significant markers, no QTL mapping involved here. So the reason we do that, again, under the assumption for a highly complex trait, quantitative traits, we have many small effect alleles. These types of traits, a genomic prediction using all the markers, oftentimes works a lot better than a marker-assisted selection model. All right, so this is a study done by Rex Bernardo's group, where they compared, I think, 36 instances of different population and trait combinations, and they found that in every single case for these quantitative traits, a genomic prediction worked better than the marker-assisted selection prediction. Okay, and you can look at several simulation papers here, such as my own, and also see the same situation where a genomic prediction model works a lot better than a marker-assisted selection model. So that's where we are, and if we look at this genomic selection paradigm, we have several questions that surround this process. Okay, first of all, which markers do we choose? How do we do the genotyping? Second of all, how do we design a training population to build the most accurate models? And thirdly, how do we allocate resources for field phenotyping when our objective changes from performing phenotypic selections to building genomic prediction models? Okay, and then of course there's always the question of which model to use. There's all kinds of different genomic prediction models out there. Is one better than the other? And how do we deal with G by E effects? These are all questions that many people have explored very thoroughly, and there's lots of good papers out there on these questions. We've come a long ways in the last 10 years or so in answering these questions, how to design an optimal genomic prediction model. But today I want to talk a little bit about this process right here, where actually can we actually implement genomic prediction in a breeding program, okay? Where are we most effective in using prediction and selection? So where to implement genomic prediction? Okay, so you'll have to suffer through a few breeding pipeline slides here. So this is a typical maize hybrid breeding pipeline, I think. Make a cross here. We take the F1, cross it to a haploid inducer, do the doubling, increase the seed from the DH, and then we do a couple rounds of test crossing. All right, so we take our DH candidates here, and we cross to, say, one elite tester, and then we do yield trials. We make a selection. We cross to several other DH testers, elite testers, make a selection, and then the remaining double haploid lines here, then we'll take those and possibly make crosses between the two heterotic groups so that we get a bunch of possible single crosses. So these are the remaining DHs, cross them together, get these single crosses. And we perform several years here of advanced yield testing and pre-commercial yield testing. Okay? And of course in maize, a hybrid system that's been established for a long, long time, we have two heterotic groups. Okay? So these things are going on in parallel in each heterotic group, and then you bring the heterotic groups together to create potential commercial products. This is just sort of a canned version of a breeding program. Of course, there's lots of nuances in any actual real breeding program. So how do we incorporate genomic prediction into such a breeding program? And I say that the easiest, most straightforward, and probably the first thing I'll do for the soybean breeding program is to use it to reduce the phenotyping. Okay? This is a very effective thing to do. You can take your DHs here that you've just created, you can genotype them. You can actually put 50% of them out in the field for top crossing and yield trial 50% of those. And then you can leave 50% of those in, say, cold storage, and then predict the rest and put those out in the field, the ones that actually are predicted to be good. All right, so you're leaving a bunch of individuals in cold storage. You're not spending the resources on those individuals to phenotype those individuals. And this works out in terms of being able to effectively test more individuals through genotyping. It'll save you money and allow you to do more testing if your genotyping is in fact cheaper than your phenotyping. All right, so what this allows you to do then is basically widen your funnel for the same amount of resources in your breeding program. There's also the option of rapid cycling, where we basically, if our genomic predictions are really, really accurate, perhaps we can make choices here on parentage very, very early in the pipeline. Okay, so maybe as soon as we double our haploids and create those haploids, we take our individuals here and we predict them. If they have a high predicted breeding value, we use those as parents right away, rather than waiting the extra 2 to 5 years to use those individuals as parents. All right, so this could increase your genetic gain over time. So in this case, of course, we're shortening the funnel. There's an example of this recently published in Nature Plants, where this doesn't actually have anything about genomic prediction in the paper, but this paper by Watson et al., I think Lee Hickey from Australia, I believe, was the corresponding author on this paper. And they showed that by manipulating growth chamber effects, they can actually take wheat plants and barley plants, and they can get these 6 generations per year, okay, by exposing them to sunlight 22 hours per day and having an increased temperature in the growth chambers. Okay, so they rapidly induce flowering and seed maturation, and then get 6 generations per year. So if you can do genomic prediction in this context, you could possibly get, maybe up to 6 generations per year of selection if you're that confident in your predictions. I mean, from my standpoint, at least right now, I'd be awfully nervous about what will come out of those growth chambers after that many cycles of recombination. Not actually having seen those things in the field, and this takes a lot of logistics to keep that level of intensity of crossing and plant generation going, generation after generation. So this is very difficult, but I think it deserves exploration, and it could possibly increase genetic gain quite rapidly over time. All right, so besides those basic applications of genomic prediction, I want to think about using genomic prediction to sort of do what I call broaden the reach of evaluation in the breeding program, okay? So I think by leveraging predictions, we can more thoroughly explore germplasm resources. We can look at all possible breeding crosses. And as I said, crossing soybean is a really difficult endeavor. It would be nice. I can't make that many crosses. I can only make in my program about 180 crosses per year, but I have thousands of potential crosses I can make. It would be nice to explore that space of all potential crosses through prediction and choose those crosses that are predicted to be the most effective in terms of creating a breeding population that has a high mean and a high variance, which is what you want. There's the issue of variety placement, and there's the issue of hybrid performance, which I'll spend most of my time on today. But I want to touch on a couple of these things before I get into the more details of hybrid performance. So, germplasm resources. Germplasm banks are being genotyped at a rapid rate. We have germplasm banks that consist of tens of thousands of accessions. These things are being genotyped en masse. The soybean collection, for example, we have 22,000 soybean accessions at Urbana. All these things have a genotype of 50,000 SNPs. All right. Not all these things have been phenotyped. There are other germplasm collections I know that I think they fully genotyped. The maize germplasm collection is symmetric, right? But they probably haven't phenotyped everybody there. So, the question is, can we use some form of prediction in those germplasm banks to enrich our selections of accessions for complex traits? So this is an example in soybean that we pursued. So we took all the genotype information on these 22,000 accessions. We used historical data collected as part of the germplasm collection and built prediction models. And you can look at the correlations, but at the end of the day, this is the one I want to look at right here, this pie chart, where if you take a random set of accessions from the collection, you'd expect, of course, you have the best half is in the top 50%, and the worst 10% is in the bottom 10%. This is just a random set. But if we enrich that random set through our selections by only choosing the best 10%, what we observed is that basically we can enrich that set for the better half up to 88% of the enriched half. And almost more importantly, we've almost eliminated those bottom feeders there, the worst 10%. Yeah? We've effectively eliminated them from our selected enriched set. All right. This is an area, variety placement, that I have not worked on, but I know Pioneer has worked on quite a bit, and some other people have worked on. I take this example here from Charlie Messina's work, published this last year. And the idea is we have all of our candidate genotypes in our breeding program. We genotype them all. We phenotype the subset, or maybe all of those. And then we combine that information with sophisticated crop growth models and environmental kinship models built through basically what Duke talked about earlier, envirotyping, right? We feed all the information into these models, and we build the genomic prediction, and then we can predict how any one individual will do in any possible scenario across the country. Yeah. So we can't possibly test all our candidate lines across all possible environments that a farmer may encounter in the future, but by combining weather data and environmental data and crop growth modeling and these environmental prediction models, we can in silico evaluate all these individuals. So I think a pretty powerful application. Still a ways to go in this, but I think it's an area worth much further exploration. All right, so fourthly here, let's look at hybrid performance in the space there. So hybrids, the reason we produce hybrids, of course, is because of heterosis, where the hybrid performs better than either parent. Of course, this is definitely the case in maize. There's lots of heterosis in maize and lots of other crops. And really, the idea of hybrid breeding goes all the way back to George Shull in 1988 when he published his paper on the composition of an open pollinated field of maize. Basically, he laid out, the pure line hybrid breeding method back then. And what he recognized were two things. He recognized that an open pollinated field of maize, which most fields of maize, or all fields of maize back then were open pollinated fields of maize. An open pollinated field of maize consisted of a complex array of hybrids. A heterogeneous complex field of hybrids. And if there's a heterogeneous mixture of hybrids, then of course they vary for their genetic value, for the traits of interest. Right? So you have variation in the genetic value for the traits of interest, namely yield in corn across that entire field. Okay, so he recognized that. And then secondly, he recognized that through multiple rounds of self-fertilization, you could reduce those hybrids down to their elementary species. Okay? Or basically, you could create pure lines, pure breeding lines through inbreeding. Homozygous individuals that which, because you created pure lines through self-fertilization, you could replicate these individuals indefinitely, right? So what he proposed is that the job of a corn breeder should be to identify and replicate the combinations of individuals that create the superior hybrid, the most superior hybrid in the field. Okay? So again, the objective of the corn breeder should be to reduce the individual down to their true breeding forms and then replicate them and then find that combination that creates that superior hybrid in that complex array of individuals in open pollinated fields of maize. So that's what you should do, but the problem is that you can't possibly evaluate all combinations of individuals over multiple environments. So if you have, let's say, just, of course, 200 individuals, if 200 individuals, you have, 40,000 possible hybrids to evaluate. That's far too many to actually evaluate. And then on top of that, this one here. So one of the first things that corn breeders did back in the early days is to sort of reduce this hybrid evaluation problem is that they formed heterotic groups. All right, so they took all the individuals and they divided them into basically two heterotic groups so that they would cut the number of individuals that they had to evaluate by quite a lot, but you still have a large number of individuals to evaluate. Even if you take those 200 individuals and you divide them into two heterotic groups here, 100 here and 100 there, you still have 10,000 possible hybrids to look at, right? So the hybrid evaluation problem remains. So over the years, there have been lots of approaches to hybrid prediction and conquering that hybrid evaluation problem. An obvious first choice would be the inbred per se performance. Just look at the value of an inbred for a certain trait and then use the value of that inbred to predict how well the resulting hybrids may perform. But for many complex traits, especially for grain yield, this has no predictive ability at all. This is absolutely pointless. Okay. Top cross test I'm going to mention in more detail here. Genetic distance based on molecular markers. I think earlier work on this has shown that you can use genetic distances to identify which individuals should go into which heterotic groups. All right. But the problem is that that's not really where corn breeders operate. They already operate on between heterotic group predictions. Okay. So the goal will be to use molecular markers to identify which individuals create superior hybrids when you already cross between heterotic groups, but that didn't turn out to be so accurate. Fourthly here, we have best linear unbiased prediction based on pedigrees. All right, so this is great. This is great for identifying individuals that have superior combinations between families. But a lot of what a corn breeder wants to do is they want to identify individuals that perform well within families. Right, so individuals within families have the same pedigree. Individuals from closely related families have similar pedigrees. So when you're talking about differentiating individuals here from the same pedigree or closely related individuals from closely related pedigrees, this has much less power. So, and then today we'll be talking about genomic prediction. Okay, so the top cross test. So we have, in a top cross test, what we have here is we have our one heterotic group, we have our second heterotic group. We take an elite tester from one of our heterotic groups, we cross it on to everybody right here, and we generate what we call top cross progenies, right, for testing. And we put those individuals out in the field, and we basically select top crosses based on that, and the individuals that do well survive to the next round of selection. All right, but the problem with the top cross test is that it can take multiple years and resources. So we have take individuals here, we cross into one tester, do yield trials, and then cross to a second tester. All right, so it can take, in my little diagram here, for whatever it's worth, it takes 2 years. Can't see from what. Yeah, and then the other problem is that, you know, it's possible that you could have a negative interaction. You could have a superior candidate inbred line that may possibly do well in combination with another individual in your breeding program, but if it happens to have a negative interaction with your first elite tester, then the individual could be incorrectly discarded early in the pipeline and never have the chance to actually be crossed to anybody else to maybe, so we can assess its full potential in hybrid combination. Okay, so the question is, can we use genomic prediction to predict all possible single crosses as soon as the candidate inbred lines are developed? All right, so I'm talking about up here at this point in the pipeline, all right? So we get our DH seed here right away, and then can we immediately right here predict how all possible combinations of these individuals would perform. So once again here, this is in another version here, a traditional phenotypic selection pipeline for hybrid breeding. We have you know, our two heterotic groups, we make crosses, we create biparental populations, we get our inbred progenies, we do a few years, 2 or 3 years of top cross testing. We have our lines remaining after the top cross test. We make all possible combinations among those individuals, more years of evaluation with hope that we have a new hybrid. This is the scheme that I have here. But is it possible to reduce that, or actually eliminate that top cross portion of the pipeline? So rather than do that, we take these individuals here from our biparental populations, so get our inbred progenies, and then predict how all individuals here will perform in combination with each other right out of the gate, essentially. Okay? And then identify those individuals that are predicted to have high single cross value and do multiple years of field evaluation for them. So put those two pipelines side by side, you can see that, you know, we're saving quite a bit of time over here. And then I don't have it illustrated right here, but you could also eliminate the possibility of these negative interactions with testers in this testing phase right here. Take a drink of water. Okay, so in terms of the details here, I'm mostly going to present the work of one of my recently graduated PhD students, Dineshwar Kadam, who I call Dinesh. And his objectives were to examine the potential of a genomic prediction on single crosses in the early stages of a hybrid breeding pipeline, okay, those DH lines that come right out of the gate, and evaluate the non-parametric genomic prediction models for early single-stage crosses, as well as look into optimizing genomic prediction models for this purpose. So this is the germplasm we use. We have 2 data sets. We had a data set from Illinois, which we got from Dr. Martin Bone there. His data set was awfully similar to a data set that we generated ourselves in Nebraska, where basically we had anywhere from 3 to 8 inbred parents from one of the 2 heterotic groups. We have the Iowa stiff stock synthetic heterotic group and the non-stiff stock heterotic group. And then from those, we created multiple biparental populations by crossing those individuals together. And then from those biparental populations, we generated anywhere from 46 to 171 random inbred lines, either random recombinant inbred lines or random double haploid lines. All right, and then we took those random inbred lines and crossed them together to create either 312 random single crosses or 481 random single crosses in the case of the Nebraska data set. All right, so this is just another version of that. So we have, we have our populations here that we create. Okay, and then from these populations, we derive a bunch of recombinant inbred lines or double haploid lines. In the case of Martin's data set, our data set was recombinant inbred lines. We do this on both sides of the heterotic pattern. And then we take those random inbred lines, cross them together to make a bunch of random single crosses. All right, so this represents, the structure of a breeding program right out of the gate, right out of the formation of their inbred progenies or double haploid lines. All right, so what we do is because we can't we don't have the resources to make all possible crosses, we use basically a incomplete factorial, pretty much made at random, just how things kind of nicked in the field and just how things worked out in the field. So you can see how we have our families here, and then each line or each row or each column represents a inbred progeny from that family. So here we have 35 columns here in this segment of the matrix, 38 rows in this segment of the matrix, and where you see a colored cell there, that means that we actually created and observed that single cross. Okay? So this means that this individual here was crossed with this individual here, and we've observed that single cross. And then, of course, all the white cells are individuals that we could have made, but we didn't have the resources to make, and just weren't made by chance. All right, so in terms of phenotyping, again, we have the 312 single crosses. We have the 481 single crosses. These things are phenotyped across 5 environments in the Illinois data set case, 4 environments in the Nebraska data set case. You see the experimental designs there listed. Traits I'm going to be looking at are grain yield and plant height. And genotyping, for both cases, we use genotyping by sequencing. You can see that at Nebraska, we're much more successful in getting many more SNPs than they were at Illinois for whatever reason. I think we just used a more advanced sequencing platform there. All right, now in terms of models. So we have basically a standard general combining ability and specific combining ability based GBLUF model. All right, so you see we have all of our fixed effects in the model, which I don't need to list out here. And then we have our GCA effects for the females and our GCA effects for the males and our SCA effects for single crosses. All right, so the important part is how we actually model these things. And yeah, that's what those are. For the GCA effects, of course, we assume them to be random. And when we assume them to be random, we can specify a covariant structure. And the covariant structure that we specified for these was a realized genomic relationship matrix calculated using the markers. Okay, so this is how we're borrowing information among individuals in our model by specifying this covariant structure. And for the SCA effects, we modeled what's known as a dominance relationship matrix. So basically, this will reflect the probability that any 2 hybrids share the same dominance deviation. All right. So we're sharing information using the markers, creating relationships, putting those into our mixed models, and sharing information among individuals. Basic GBLUF model and genomic prediction. Okay, so first things first. First question should be, how did our genomic predictions perform compared to a top cross test? And the unfortunate thing is that we didn't actually perform a standard top cross test alongside this experiment, so we can't make that simple comparison. But we mined the data in such a way that we're able to sort of mimic the top cross test. Okay, so we took one individual, we looked how it performed when crossed to one other individual, sort of assigned that individual as a tester in our data set. And we did that for every individual in our data set. So we're doing what I call a mimicked top cross test, using the data, mining the data. And you can see that, if you are okay with that explanation, you can see that the methods here our genomic predictions based on the genome here did quite a lot better than our mimic top cross test. All right, so borrowing the information across all the individuals in our population through the genome is doing a lot better on predicting the ultimate value of that hybrid than by predicting just based on one one single tester, all right, simulated tester in our data set. And then if we actually estimate the phenotypic-based GCAs, okay, sum those together based on all the phenotypic data, we do about the same, all right, as our genomic predictions. So the take-home message here is that our genomic predictions do about as well as our phenotype-based GCA estimates. All right, so the point is they're doing quite well. All right, so can't really see that very well. The gray is kind of faded out there, but anyway. So the other question here is, oh, there it is. Okay. What does it look like when we look at tested parents versus untested parents? So we have an individual in our breeding population. We've either tested it by crossing it and putting it in the field, or it's completely untested, hasn't been observed at all. All right, or it's been tested based on one individual to either as a male or tested once as a female. All right. The parents were single crosses. So the T2 situation here, you can see that our prediction accuracies are actually quite higher. When I say prediction accuracy, I mean it's a correlation between the prediction and the observation. All right, and these are all based on cross-validation. As Sindhu explained earlier this morning, everything is calculated using the appropriate cross-validation techniques. So of course we have much higher prediction accuracies when we observe both parents in the field. For a single cross for which we observe neither parent, we have much lower prediction accuracies. But they're still not terrible, but not as good as I would like. Yeah, and of course they're intermediates when you look at just one parent, right, for grain yield. For plant height, they're all pretty high, but grain yield is what we're really interested in here. Okay, and so then what about models? I kind of glossed over models before, but the thing about implementing genomic prediction in the breeding population is that we oftentimes have this, or almost always have this problem of the small n and large p. Okay, so we have many more variables to estimate the effects of than we have observations. All right, so we can't use just a typical least squares type of approach because we don't have enough degrees of freedom to estimate the effects of all the things in our model simultaneously. All right, even if we did have enough degrees of freedom, when we have this large a p, we're going to have all kinds of problems with multi-collinearity and overfitting in our model. Right, so we can't use those models we normally use or used in the past. So we use all these other models that have been developed. And there's a whole array of genomic prediction models out there that have been used. All kinds of Bayesian models, non-parametric models, and as well as just mixed models. Okay? So I already mentioned GBLUP, and its mathematically equivalent model, RRBLUP, okay, are the most commonly used genomic prediction models out there. So just real simply here, to explain genomic prediction, genomic best linear unbiased prediction. Essentially, again, as I said before, we're essentially sharing information across individuals here using the realized genomic relationships. All right, so what we do here is we have this effect in our model here that we call the polygenic effect. We fit it as a random effect, and we fit it as a random effect. We specify its covariance structure here as the genomic relationship matrix. All right, and so this is what this matrix looks like right here, and I don't know why these M's and O's and L's are in here. Some sort of issue with my formatting on my slide, but ignore those. So just look at the G's. These are supposed to be G's in here. All right, so these are just basically these off-diagonal elements are here, basically just the realized genomic relationships here between all the pairs of individuals in our population calculated using our marker data set. All right, so the same situation would look like if we just use this for pedigree BLUP, but if we use pedigrees, of course, those would be coefficients of co-ancestry based on pedigree data. These are genomic realized relationship matrix estimates. So, of course, individuals in our population here vary for their relationships. And of course, individuals vary within families for their relationships. And we're making the simple connection here that, of course, individuals that share higher proportions of their genome. This is Jim Holland gave this great example one time at a meeting I attended, so I'm going to take a page out of his book right here. Individuals that share more of their genome resemble each other more than individuals that are less closely related or more distantly related do. So they resemble each other less. Then, of course, there are individuals here that are, from the same family, but they may look quite unlike each other, even though they're actually, you probably all have siblings that, maybe look like more like one sibling, they can look like another sibling, and that's the Mendelian sampling term, right? There's differences in the realized relationships. So that's what we're trying to capture here with this genomic prediction GBLUP model. All right, so the thing is that there's lots of, I looked at a lot of different models there. We have a lot of alternative models to look at in genomic prediction, and we can compare those to a GBLUP prediction accuracy. Okay, this is sort of the null hypothesis, I would say, or the standard model. This is the one we want to compare it against. And you can see that, if you look at actually these comparisons are in soybean. So you can do this for lots of crops. And you can see that they all pretty much line up on this one-to-one line. Okay, there's a little bit of deviation here, but if you're within this yellow box here, I would say this is sort of within the who cares zone. Okay, so if you're dissimilar to each other, it doesn't really matter so much. You're talking about just 1 point or 2 points in difference. All right, so it's not really maybe it's statistically significant in some cases if you have enough data, but practically speaking, it's not really too big of a deal. So GBLUP works really well. It's a really, really robust model. GBLUP/RBLUP work quite well. All right, but then we have all these new models out there that's machine learning models that are comprised of things like simple neural networks, deep learning networks, support vector machines, all these types of things. And we saw some good examples today of how powerful these types of models are for processing image data and for high throughput phenotyping. All right, so how do these things work for genomic prediction? So we looked at these models for this single cross hybrid prediction case. All right, so we looked at reproducing curval Hilbert spaces, which is a non-parametric or semi-parametric model. We have support vector machines and neural networks. Okay, so we looked at each of these models in terms of predicting single cross performance, and we found pretty much what we found in other cases where there's really not much of an advantage to using these models in this scenario. Okay, so again, GBLUP is a standard GBLUP is a very robust model. That said, though, however, we have a fairly shallow data set here. Okay, so we had a really, really extensive, really, really deep data set, mining tremendous amount of historical data, then perhaps the situation would change. So what about the correlations of the actual GCA effects and the SCA effects? Okay, so here we have we basically separated from our hybrid performance, we separated our GCA effects out from our SCA effects. Okay, so now we're looking at, okay, how well can we predict the GCA effects versus how well can we predict the SCA effects, or those effects related to the specific combinations of hybrids. Okay, which is going to be mostly non-additive variants. So you can see that in all these cases here, for all these models, we predict our GCA effects quite well. All right, but none of these models, including the non-parametric models, which are supposed to be so great for modeling non-additive variants, predict the SCA variants at all. Okay, I mean, there's a couple reasons for this. One reason is that we just didn't have that much SCA variance in this data set. There's a good reason for why these aren't really that predictive. Okay, so most of our prediction accuracy from these models is basically just coming from being able to predict our GCA effects, which GBLUP is very, very good at in this data set. Okay, so finally, the question of how much time do I have? Do I have any time? 0? Okay, all right. Okay, so this question of training population optimization. So the assumption here is that not all training populations are equal in terms of the relevance for a breeding program or for a candidate set that we want to predict, or the information content that they contain. All right, so what we want to do then is we have a possibility of a training population. We have a target population. We can feed information from these populations into optimization algorithms. Okay, pick out a best training population, phenotype those, and if our training population is actually optimized, our prediction accuracies will exceed those of a randomly selected training population. Okay? So this has been demonstrated several times in the literature in several papers. You can take these optimization functions out here. This is the one that optimizes the prediction error variance that comes out of the mixed models. This optimizes something called the coefficient of determination, which is a function of the prediction error variance and the genetic variance of your possible training population. You combine these with optimization algorithms, such as a simple exchange algorithm. Okay, if you do all this, you'll get more informative training populations. All right, so it's a way for you to select a smaller training set that has a higher prediction accuracy. Okay, so how well does it work in this data set that we have right here? So what we found is that it works quite well. So for grain yield, we can take a training set here among these single crosses, and we can optimize it based on these optimization functions or these objective criteria. And we can find training populations of only 100 individuals here that do as well as randomly selected training populations consisting of 400. All right, so we're saving a lot of resources here in terms of phenotyping by optimizing our training populations. And then secondly here, for this training optimization exercise, we only use the observed single crosses. Okay, we only looked at those 481 individuals we actually made and actually put in a field. Okay, so we can do that. But what happens if we use everybody? Okay, I'm talking about the whole space here right now. You make some set of crosses here between some individuals. You have a few they actually made. You have a whole universe of individuals you didn't make. You can look at those individuals in silico because you have each genotype, and you can in silico create each individual hybrid. All right, so what happens if we look at the entire universe of all possible single crosses we could have made? All right, so here, again, optimization based on all 481 observed single crosses, optimization based on all possible nearly 10,000 single crosses. And what I want you to see here is that we get a pretty good bang here when we optimize based on all possible single crosses. All right, so these lines are about the same. You can see that these lines converge here when we only have 481. But if we look at all possible, the lines are separated by quite a bit. I don't actually have this quantified in my mind right now, but it's it's a pretty good boost. It's a heck of a lot of a bigger boost than comparing different types of models. All right. So then we look at all possible single crosses here, and all these are all the possible ones in terms of their predicted values. And the point here is that you can find 50 untested, unobserved single crosses that had higher genomic prediction values than anything we would actually observe. All right, so if I were a corn breeder, what I would do is I would employ this, predict all possible single crosses in my breeding program, and I would definitely, if I'm not fully implementing these methods in my program, I would at the very least look at these 50, put them in the field, because they may be better than anything I actually happen to make by chance, or maybe something better that actually survived my test crossing pipeline, maybe. So these are my conclusions. We'll broaden the reach of evaluation. I think there's a lot of potential for this in optimizing breeding programs, and training population optimization holds a lot of potential. Yeah. With that, I'd like to thank the people involved in the study, especially Dineshwar Kadam, who graduated just recently, Dinesh's committee, Martin Bone and Sarah Potts for sharing their data and working with us on the Illinois data set, and Ron from Pioneer, and then my lab that worked on this data in Nebraska. So with that, thank you.
Thank you, Dr. Lorenz, for the talk. So in the interest of time, we'll probably entertain 2 questions.
Okay, thank you for the work. My question is that your genotype inbred the parents or the test cross F1? Top cross?
Parents.
Parents.
We have the parents. The parents are true breeding lines, so we genotype the parents. We can in silico genotype all the hybrids.
You genotype the parents?
Yeah, the parents, yeah.
How many markers you use?
Well, in the one case we had 2,000, in the other case we had 23,000. But it was GBS data, so we had 23,000 marker data points, but a whole lot of them are missing. I can't remember the exact number, but it was something like 30 to 50% missing data, so we had to impute the missing data points.
We got a question. Okay, thank you, Dr. Lorenz. One more question from Dr. Singh.
This is a question that both you and Dr. Hemmert can answer. In his presentation, Dr. Hemmert mentioned a yield of 542 bushels per acre of maize. Is that, if it is correct, this is like 30 tons per hectare of grain. And if we take 50% harvest index, another 30 ton is biomass, which is like 60 tons per hectare in 5 months. This is like theoretical estimated yield that can come out if we get 100% efficiency. So my two questions: are you examining this hybrid, why it is happening? Maybe genotype the invert lines and we use this as an elite genetic improvement population, so we can stabilize this. Secondly, have we reached the theoretical base that we stabilize this? The global yield, average yield of corn is 3.8 tons per hectare. US average is 10 tons. Here it is 30 tons. If we can raise the world yield potential even to 10, so have we learned something? How are we going to make use of this elite variety that gives us 30 tons? And I think we can use all the genetic data, genomic data, and so on to understand this particular hybrid and go from there. Thank you.
So I think that 530 bushels or so, that was a yield contest winner, right? So most of that's environmental. I'm guessing that they chose, did they have a Pioneer hybrid? They did, okay. So they had a good hybrid. So some of it's hybrid, but a tremendous amount of that is going to be the environmental, a lot of optimum fertilizer application, irrigation, all that kind of stuff. So, but I do agree that it seems to be that there's these individuals that sort of pop out of these breeding programs that are heads and tails above everybody else. And you know, I guess, I don't know, you guys looking at those hybrids in your program, the ones that sort of...
Yeah, you're right. The environment management component is huge there for those theoretical maxima. But one thing we've done to try to understand why they're yielding so much is the work that Charlie Messina's group has done with crop growth models. Interrogate those to understand what the plant is doing physiologically in those target environments. Yeah.
With the sample size of one or just a few, it's really hard to look at the genome because you have
What
so many.
I was driving at is, can we take the genotype of that hybrid, two inbred lines that are making up whatever it is, and go deep into genomics and see that is what will be an elite genetic material for future work. Because 30 tons grain and 30 tons biomass is a lot of it. I think it is almost like theoretical model. So we should use all our knowledge of genomics and all these genomic selections and so on, use that population as a base. Are other related lines which are not 540, but 400 and above. See, 150 bushels per acre is like 10 tons. So anything 150, 10 tons, another 150, 10 tons, we are talking about 30 tons. So can we take some of those elite hybrids and focus on them with our knowledge of, sequencing and genomics and all those things that we are talking about? That will solve our problem. If world average of 3.8 tons can be raised even to 6 tons, we have fed all the world. Thank you.
So one of the things we're doing as we move into an editing approach is we need to really understand the genotypes that we're working with. So we definitely are building data sets internally that elucidate all those details, certainly for important hybrids like that.
I would like to give a final reminder that the social tonight is at Cafe Excel and it starts at 6. And with that, I'll hand over to Amani.
We're going to have a short announcement from Dr. Don Jones.
Hi. I want to say a shout out both to the speakers and to the organizing committee for the the session today. It's been excellent. It is a lot of work to put these on. So let's have a round of applause again for the group and the speakers. I'm here to mention this Borlaug Scholars brochure that's on the table for the graduate students and undergraduates. This was kindly thought of by Don Cummings, retired breeder from Monsanto that's been involved with NAPB. And read through this brochure on the inside cover here. There's some links over here for both the undergraduate and graduate students. And this is support to attend the NAPB meeting. This year it's in August, first week of August, or I think August 7th through 10th in Guelph. And I encourage you, if you can attend, because for plant breeders, it's one of the better meetings I think is out there. So appreciate it.
Thank you, Dr. Jones. We will have a last few minutes. We will have Dr. Bogsberger give a few closing remarks.
Wow. Thank you for all of the stimulating participation. Thank you to our many sponsors, especially thank you to the coordinating group from this. We really, really appreciate the efforts that the students have put in. Let's give everybody a big hand. On behalf of the Soil and Crop Sciences Department, we are really proud of this group. We are really pleased with what our students are doing. We are pleased with the participation and the interaction that's going along across the schools. We really, really think that the future is bright for plant breeding and genetics and omics. And so thank you again for your participation. Enjoy tonight at the social, because that's your opportunity to really get things cleared up and find new opportunities that can be explored together. Thank you.
So I would like to mention that though we are at an end today, we still have one more event scheduled tomorrow. We will be having a panel discussion in Heat 440 where some of our speakers are going to be there and we can continue this discussion for a whole hour. We will be serving pizza, so those of you that can make it, I would encourage you to attend it. So as we end this fabulous event today, I will take a few minutes of your time to thank all those people responsible for helping us put together such a big event. So first and foremost, DuPont Pioneer, which continues to contribute to organizing such a big event. We are honored to be a part of your event and are grateful for all the opportunities we get from organizing this. Thank you. In addition to DuPont, we also have other sponsors at Texas A&M. The different departments, starting with Soil and Crop Sciences, the Institute for Genome Science and Society, Biology, Horticulture, Molecular and Environmental Plant Sciences, Biological and Agricultural Engineering, and Plant Pathology. We've also received support from the College of Science and Engineering of Texas A&M System's Corpus Christi location. We've also continued to receive support from the Texas Peanut Producers Board. All of you who received a T-shirt today, it was donated by Carton Incorporated. Some of the beverages served today were donated by Pepsi Cola. In addition to these departments and organizations, we would like to thank some of the individuals that helped us in organizing this event, starting with Leanne Haig, Jim Lukeman, Dr. Wayne Smith, Kathleen Peacock, Joni Suwick, Deborah Sutherland, Beth Ludeker, and Taylor Barfield from the Department of Soil and Crop Sciences. These individuals have helped us behind the scenes on finances, borrowing equipment, and generally giving us some good advice. I would also like to thank the PIs of all the committee members, because you have allowed us to use some of our work hours to do something that was not research, and we appreciate your patience with us. I would also like to thank all the speakers of today's event for taking your time to share with us your research. Thank you very much. So we would also like to thank all the volunteers who have shown tremendous Aggie spirit today by donating their time in helping us organize such a large event. Without your help, we would not have pulled it off. I would like to remind everybody that we usually send out an evaluation after the event. So I would request you to take 5 minutes of your time to tell us how we did and tell us what we can do to improve. All of these suggestions from previous years have helped us improve our event. So as mentioned earlier, the Plant Breeding Symposium is part of the DuPont Pioneer Symposia series, and we have several such events happening across the United States and throughout the world. Most of these events stream their webinar, much like our own. So we would request you to keep track of them and attend all the interesting talks through webinar. If you missed one of today's presentations and you want to re-watch a last year's presentation, we will be posting these in our YouTube channel for those which we have consent to record, and they will also be available on our website. One last reminder that we have a social at 6 p.m. at Cafe Excel. Everybody is invited along with your significant others. We will have free appetizers and a cash bar. It will be a great opportunity to network, so I highly recommend you to attend it and make it fun today. So lastly and most importantly, I would like to thank all of you for sticking with us till the very end. This concludes the 2018 Texas A&M Plant Breeding Symposium, the Omics Era, a new page in plant breeding. Thank you very much. For those on the webinar, the symposium has now ended. Thank you.